Okay, please, can we all see my screen now?
Yes.
Okay, thank you very much. Okay, yeah. So today we're starting with we're starting our microbiology today, and we're starting with the basic bacteriology.
For my father, I can't see your screen, please. So.
Ah, but I thought somebody said they can see my screen now.
Yes, I can see your screen. I don't know. I can
Dr.
see
Paul,
it.
it's most likely your network. We can see your screen, Dr. Paul.
Okay, thank you, Dr. Precious. Dr. Paulette, maybe you should try and log out and log in again. I think should come. Dr. Paulette, are you doing that?
It will not disrupt the class.
Okay. Hello?
Okay, don't worry. I can see your screen now.
Okay. So, um, first of all, microbiology is a little bit different from the other subjects. Again, hello.
Okay.
Some background noise. Like, uh, sorry. Okay. Yeah. So, it's very different because we don't have much principles here that we can use to explain, bulk of what we are talking about. Though we'll still try as much as possible to give some concept to understanding some of them. But microbiology will require some little memorization. I'll be very sincere with us. Yes, that's microbiology, and of course we are familiar with it from right from medical school. So without wasting time, let's just go straight to the stuff. So we are starting with the bacteria today. So we are going to talk about the structure of the bacteria, the components, and their function. The the typical bacterial cell, what it's made up of, and the function of each of those structure. So it's been categorized into three parts: the appendages, the specialized structures, and the cell envelope. So let's start with the appendages. From the word appendage, it means um, additional structure, like external structure that is attached to a particular cell. So it might be there, it might not be there, depending on the cell. Some cells have it, some cells don't have it. And what are the two major or three major appendages that we are concerned with? The flagellum, the pilus, and the fimbriae. The flagellum, the pilus, and the fimbriae. So the flagellum is made up of only protein. So the flagellum is made up of only protein. It's just like a whip-like attachment to the bacterial cell. And what it's responsible for is just to ensure motility. It allows the cell to move from one part to another. It is not all bacterial cells that have flagellum attached to their surface. So any bacterial cell that has flagellum attached to the surface will be motile. Anyone that doesn't have the flagellum would be non-motile. So what's the chemical composition of the flagellum? Protein. That is all. But before I move, with respect to our knowledge of immunology, if you guys can remember, I'm very sorry, those of us that didn't join us for immunology course. Uh, I just want to throw more light about some aspect of the immunology that we talked about there. So if you guys remember when we were talking about the pathogen natural molecular pattern, I don't know if you guys remember that part. That we are saying there are some pathogen natural molecular pattern that the innate immune system tends to recognize in a microbe before it mounts immune response towards it. An example is the herpes for gram negatives. An example is the flagellum. An example is nucleic acid for viruses. So those components of the bacteria or the virus can be recognized by the innate immune system, and they want to mount an immune response to attack it. So flagellum or flagellin is an example of a pathogen associated molecular pattern. That is a molecular pattern that is associated with a pathogen. What's the pathogen? The bacteria. What's the molecular pattern? That is anything that can be recognized by the immune system. What is that? The flagellum. It can be any other thing. We are still going to talk about those other ones, but I just want you guys to pay attention to the fact that the flagellum itself can be recognized by the innate immune system and they can respond to the bacteria because of that flagellum. So flagellum is strictly for motility. The other appendage that we have, the pillars and the fimbriae. The pillars and the fimbriae. Both of them are classified together because they consist of the same chemical composition. They both contain glycoprotein, not like flagellum that is only strictly protein. These guys, the pillars and the fimbriae have glycoprotein. That is, in addition to the protein component, they have carbohydrate attached to it, glycoprotein. What's the function of this appendage? So the pillars is just for conjugation. The pillars is essential for exchanging genetic materials between bacteria. That we are still going to talk about bacterial genetics and we'll emphasize conjugation. So you guys should just take it as exchanging genetic information. So if one bacteria has a genetic makeup that is favorable for another bacteria, maybe antibiotic resistance or ability to use some certain metabolites, it might want to share that gene with another one, with another bacteria that is devoid of that attribute. How does it go about it? They mate together by, or they connect together with the head of the pillars. So you say sex pillars for conjugation, which allows for genetic transfer. So after the pillars, which other one? The fimbriae. So fimbriae is more like an attachment appendage. So it's basically there to allow the bacterial cell attached to different surfaces. And the one that is very clinically significant is E. coli. So E. coli has the fimbriae, and it is the reason why E. coli can cause UTI, because with the aid of the fimbriae, it can attach to the epithelial surface of the genitourinary system and cause UTI. So the reason why it's getting attached to that epithelial surface is because it has the fimbriae. So going back, the flagellum contains protein for movement. The pillars and the fimbriae, they are all made up of glycoprotein. And what's the function of the pillars? For sexual conjugation, allowing for transfer of genetic material. What's the function of the fimbriae? Attachment to surfaces. Attachment to surface. An example of the one I gave is E. coli causing UTI, because with the fimbriae, E. coli can attach to the epithelial surface, epithelial cells
The
lining
only one. Hey.
the genitourinary system,
Okay,
causing
Dr.
UTI.
Bavale, we just lost you for like 30 seconds to 1 minute.
I'm very sorry.
Let
I
me
had a
see
call.
the
I had a call. I'm so sorry. What
one.
about?
That's all right.
Okay.
Okay. So do you guys get the function of the fimbriae now and the pillars? You guys get them now. So that is that about the fimbriae, the pillars, and the flagellum. So we'll go to the specialized structures. So specialized structures are the example of specialized structure that we're talking about here is the spore. So spore is a specialized structure because it is not all the bacteria that even use the spore. The only type of bacteria that has a spore are the gram-positive, and it's not even all of them. It is not all of them. We are still going to mention some of them during the course of the discussion. So we should just know that spore is a specialized structure and it only exists in gram-positive. So I look at the spore as a form that the bacteria tends to assume in order to adapt to environmental situations that are very, very unfavorable. For example, if the bacteria is outside the host and is within the soil, and there you don't have water, there's too much heat, it can get desiccated. You can have denaturation of the denaturization of the denaturing of the DNA. You know, the bacteria has to protect itself from that. The bacteria has to protect itself from that. How does it go about it? It assumes the spore form. So in that spore form, it's protected from harsh environmental situations. It's metabolically inactive, so it's not doing anything. It's just remaining inert, so that it doesn't die, so that it can survive until the situation becomes favorable. There's now water, they have more nutrients, they can now change back to their vegetative form. So I have a picture here to
Hi,
show us.
sorry, Babawale. Can anybody still see the screen? Okay, I can see the screen now.
Can you see, Dr. Ken? Okay. So
All right.
so this is, can you guys see this image I just tried to put beside my screen? Can you guys appreciate this image?
Yes.
Okay, so this is the image that is going to guide us as regards knowing the composition of the spore. So this is how the spore looks like, just like this oval shape. So it has the outer layer that is made up, that's called the exosporium. So that one is not very important because it's not stated here in our first stage. It's not part of what was listed here. So, but you should not know that there's an outer covering that we call the exosporium. The next one is the coat. So I'm so sorry. Let me scroll back. So there's a coat. You can see the nest lining, the coat. Now that coat is made up of keratin-like material. So after the exosporium, we move next to the coat, which is made up of keratin-like structure, keratin-like coat. Then after the coat, you have the cell wall. You can see the next layer. This one. I hope you guys can appreciate my cursor. We can see those three lines. The first line is the exosporium. The second line is a coat that contains keratin-like material. Then the third one is the spore wall or the cell wall. Now that cell wall is made up of peptidoglycan. We are still going to talk about peptidoglycan as we talk about the cell wall down when we get to the cell envelope. So typical bacterial cell wall is made up of peptidoglycan. So this spore form too, the wall of the spore is made up of peptidoglycan. Then the one that is now enmeshed, the one that is in the inner part, that's the DNA. Of course, the bacteria will have a genomic material. Mhm. So the DNA is the one that is most inside. And what's surrounding, although this picture is not showing it, I have another image, but it's not here. Maybe I'll send it to the group later. What is surrounding that DNA is dipicholinic acid. So dipicholinic acid forms like more like a covering over that DNA, investing on it directly so that it can protect it from damage from heat majorly. You see, all these other layers, they're actually trying to protect it from water, from dehydration, from destruction from chemicals, as well as heat. But the one that actually participates in protection from heat the most is actually that dipicholinic acid that is surrounding the DNA. So let's go back to what we have. In our first stage, they said spore contains keratin-like coat, dipicholinic acid, peptidoglycan, and DNA. So it makes sense to us. We can go from outward to inward now. So we said the one that is most outward is the outer covering, which is the exosporium. Then next to that exosporium is the coat, the keratin-like coat. Then next to that coat is the cell wall that is made up of peptidoglycan. Then the one that circles the DNA directly is dipicholinic acid. Then we now have the DNA inside. So that explains the whole composition. And that form can only be assumed by gram positives. And the purpose of assuming that spore form is to protect that bacteria from dehydration, from heat, or chemical damage. And we said the part that actually protects more from the heat is the dipicholinic acid that surrounds that DNA. So gram positives like Clostridium, like Bacillus, they typically exist in this spore form. So they just stay in the soil as spore form. They stay inside the soil. In the soil, you are exposed to the environment, heat, everything, no water. You can even get exposed to chemical and all those things. So instead of the bacteria to just make itself vulnerable to all those things, it will just assume that spore form. The DNA will be protected by all these coverings that we have talked about in the spore so that it doesn't get killed off. But when the spore is now inhaled by humans, it gets into the body, the conditions become more favorable. It can now change from that spore form to vegetative form that will now cause infection. You don't cause infection with spore form. It has to change to the vegetative form that's capable of causing infection. So that's a specialized structure, spore form, only in gram positives. We've talked about the composition. So don't forget, you can see it. I've seen it in one of the NBMEs. They've tested one of these compositions. You must always remember outward exosporon, then the next thing is the coat, which is the keratin-like coat, then the cell wall, which is made up of peptidoglycan, then the next thing that is covering the DNA, which is our what? Dipicholinic acid. So let's go to the cell envelope. The cell envelope is made up of multiple layers. So we are going from outward to inward. So outward to inward for the cell envelope. So the one that is most outward is the capsule. This capsule too is not present in all the bacteria. Any bacteria that has capsule, we call it encapsulated bacteria. The ones that don't have, they are non-encapsulated. And it might even interest you guys to know that there are some particular specific bacteria that you have some strain that don't have capsule, some strain that have, for example, Haemophilus. So Haemophilus influenzae have some strain that have capsule. They have some strain that don't have capsule. I don't know if you guys have come across the term non-typable Haemophilus. When you say non-typable, that is a non-encapsulated type. That's the type that you cannot classify into serotypes. But the encapsulated form, you can use the differences of the capsular composition to separate it into different serotypes. Serotype 1, serotype 2, 3, depending on the composition of the capsule. So that is that. So what is the function? What is the composition of this capsule? Please forget everything that is here, discrete layer, blah blah. What you need to take home here is the fact that the capsule is made up of polysaccharide. That is it. In fact, the way me I like to say it, I say polysaccharide capsule. I don't say capsule. You will see it in the text. They emphasize it like that. Polysaccharide capsule. Polysaccharide capsule. Well, some might really contain protein, fine. But me, I would rather stick to the fact that to the generalization that majority of the capsules are made up of polysaccharide. Polysaccharide. This is very, very important. And we are going to talk about it more again as we move down. So what's the function of the capsule? Any bacteria that has capsule, any bacteria that is encapsulated is protected against phagocytosis. Your macrophages, your neutrophils, they have no power over that kind of microbes. In fact, they have to work more. They have to work hard, to destroy that kind of microbe. Because of that capsule, it makes them virulent. It doesn't make them pathogenic. It makes them virulent. There's a difference between virulence and pathogenicity. So when you say virulence, it's an attribute of a bacteria that makes it evade immune response. That is it. But an attribute that makes it cause infection, damage, is a pathogenic factor. But a factor that makes it evade immune response is just there, running away from all those white blood cells. That particular factor will be a virulent factor. It's not actually responsible for it causing infection, causing headache, causing abdominal pain, no. But it's just making it stay within that body unharmed. That's virulent. So the capsule is a virulent factor because it makes that bacteria safe, protected from the phagocyte, so they cannot phagocytosis and destroy it. So what's the composition of the capsule? Polysaccharide, not protein, because it's very, very clinically significant. Polysaccharide, not protein. So after the capsule, what is next? The slime layer or the S layer. So S layer too, just like the capsule, is made up of polysaccharide. What does it do? The S layer, the slime layer, is just to form slime, to form what we call biofilm. So we have it here, biofilm, biofilm. So I have the definition of a biofilm here. So biofilm is an assembly of surface adhered microbial cell enclosed within an extracellular matrix. Yes. So biofilms are just like colonies of bacteria that they kind of adhere together, and they form colonies and attach to a surface. Most of the surfaces, although some of them can be endogenous, most of them are actually endogenous, but some of them are from foreign substances. Maybe somebody uses a prosthesis, or somebody takes in maybe a contact lens, or you impute a catheter into somebody. So microbes can colonize those structures and they can form biofilms within them. So they aggregate within those surfaces, and what fixes them to that surface are extracellular matrix material, so that they will just stay there and be causing damage. Example is somebody having prosthetic adverb and the person is having infective endocarditis. That is it. So the microbes will colonize that prosthetic adverb, they will form the biofilm, all of them will aggregate together in colonies. They will now enmesh themselves to that valve with extracellular matrix material. They will adhere to that valve and be causing the inflammatory response until there's eventual damage of that bag. So any bacteria that is capable of forming biofilm is not too good because number one, they have high potential of causing serious damage, and number two, there's high tendency that antibiotics might not be able to reach that area to eradicate that bacteria. That is that about the biofilm. So biofilm formation, the component of the bacteria that is responsible for forming that biofilm is the slime layer, the S layer. They are there to surface, which can be different things. It can be contact lens, it can be catheters, it can be prosthetic hip implants, prosthetic heart valve, ventilators, so many things, so many things. So that's that. Just like capsule, they are made up of polysaccharides. Capsule is to evade phagocytosis. The slime layer is to form biofilms. So the next one is the outer membrane. Now, outer membrane is only present in gram negatives, just like spore is only in gram positives. Outer membrane is only present in gram negatives. What I just want us to always remember about this outer membrane is actually the composition of the outer leaflet, because the outer membrane is divided into three portions: the outer leaflet, the inner leaflet, and the area that is sandwiched in between them, which contains the embedded protein. So we have an image. So we have an image downward, so we can see. So the one on the left is the gram positive. The one on the right is the gram negative. So we can see the outer membrane here. Can you guys appreciate the outer membrane from this arrow? Can we see the outer membrane? Hello?
Yes, yes.
Okay. So we can see the outer membrane. So there's an outer leaflet. We can see this red portion, this red one, not the pink one. No, that's the outer one. We can see the protein in between, which is the porine. Then we can see the inner leaflet. So the one that is okay, so this phospholipid. So you guys look at this pink one. You can see this pink head. Can you guys see this pink head?
Yes, the inner one.
Yes, the inner one. You can see that it is spanning. It's like it's spanning across the entire membrane. So it's actually
Yes.
coming from inward to outward. So
Okay.
those are the phospholipids. That's the way a typical phospholipid stuff is arranged. So you see the head facing the opposite direction, while the tail come together. So the phospholipid one from the inner leaflet. You can see the protein in between them, and the one that is outward is the red one. You can see the red one. You can see that red one is not spanning through. It's only present outward. But the pink one is the phospholipid head, which is spanning both inward and outward, because it's coming from inward. That's why they are calling it, they are referring to it as majorly from the inner leaflet. Well, it's not very important. The take home there for you guys to always remember is actually the composition of the outer leaflet, which is this red one that we have explained. So let's go back. It's fine if you guys, okay, so let me just, okay, so the outer leaflet, the embedded protein, and the inner leaflet. The inner leaflet contains phospholipids. We have seen the phospholipid. The pink part is the phospholipid, the one that is spanning across. The protein is the one that is enmeshed in between. Then the one that is most outward is the outer leaflet that contains this red area. So
So, Dr. Bali, is
yeah,
the inner is mostly basically phospholipids, while the
phospholipid.
outside is both phospholipids and the endotox, the lipopolysaccharide.
Yeah. So let's just take it like that. You get? Yeah. Let's just take it like that. At least from what we are seeing in the image. So, but the take home there is that the outer leaflet contains lipopolysaccharide, and this lipopolysaccharide is made up of it's made up of both lipopolysaccharide and lipooligosaccharide. It's very important we know that distinction: lipopolysaccharide and lipooligosaccharide. So what is the composition of the lipopolysaccharide, for example? So from the name, lipopolysaccharide, lipo, lipid. So it contains lipid A, lipid A. Then we are left with the polysaccharide. Now, the remaining component includes O polysaccharide and O antigen. So there's the O antigen and there's the O polysaccharide, which the lipid A. That's a component of lipopolysaccharide, which makes up the outer leaflet of the outer membrane. So for the lipo-oligosaccharide, it is just lipid A. Instead of O polysaccharide, we now have O oligosaccharide. It means that there's a carbohydrate moiety, but with a very short sugar chain. Sorry, it's a little bit noisy. Then,
Dr. Water, please, I'm lost. I don't know.
okay, Dr. Antonia, where did you get
Hmm?
confused? So where?
He said something oligosaccharide, something
Okay,
O
yes.
antigen or something.
Okay, so please, guys, let us delete that image that we have seen first, because it can be confusing. Let's just imagine that we have an outer membrane. That outer membrane has three portions: the one that is outwards, the one that is in between, and the one that is inwards. Now, that one that is outwards, we call it the outer leaflet. What is the composition of that outer leaflet? It can be two things. It can be either lipopolysaccharide or lipo-oligosaccharide, depending on the bacteria. Yes, depending on the bacteria. But you must not forget that it is only gram negatives that contains outer membrane. Gram positive don't have it. So some gram negatives have lipopolysaccharide forming the outer leaflet of the outer membrane. Some gram negative have uh, lipo-oligosaccharide forming the outer leaflet. So what is the composition of the lipopolysaccharide? Lipopolysaccharide, from the name, lipopolysaccharide. Lipo stands for lipid, so it contains lipid A plus O polysaccharide plus O antigen. You get that, Dr. Antonia?
Contains lipids, O and A antigen and O
Yes.
antigen.
Lipid A, O polysaccharide, and O antigen. That's the composition of the lipopolysaccharide. The same thing for lipo-oligosaccharide. Lipid A, but instead of O polysaccharide, O oligosaccharide and the O antigen. That's the three. It's important we remember that, because the only gram-negative bacteria that contains lipo-oligosaccharide is Neisseria. So they will ask, what is the composition of the outer leaflet for Neisseria? You will not go and pick LPS like the other typical gram-negative. That of Neisseria is lipo-oligosaccharide. Now let's now break down that composition of that lipopolysaccharide or the lipopolysaccharide or the lipo-oligosaccharide. The one of interest is actually lipid A. Yes, because lipid A is the one that can induce immunogenic reaction. That's the one that can provoke inflammatory response. You can make the white blood cells start elaborating all those pro-inflammatory cytokines, which can culminate into shock. So lipid A is an endotoxin. Yes, lipid A is an endotoxin. If you guys go back to our immunology, you will see it. Lipopolysaccharide is one of the pathogen-associated molecular patterns, just like flagellin, because it can also induce inflammatory response. I hope we remember our toll-like receptors. Do you remember the toll-like receptors?
Yes.
Yes. So what are the toll-like receptors looking for?
The pump.
The
The
palm.
PAM.
Palms.
Yes. They are looking for the palms. Example of a palm is flagellin. Example of a palm is what? Lipid A in our
And
lipopolysaccharide. Yes. We are not going to be talking about the molecular details now, maybe some other time. But we should know that once that toll-like receptor on the macrophages recognizes that lipid A, they are going to start elaborating some inflammatory cytokines like TNF alpha, interleukin 1, interleukin 6, that will now start causing vasodilation, increasing vascular permeability, and causing all those things that will now lead to septic or endotoxic shock.
Chief Bawale will put me again.
Hello?
Yeah, I said we lost you again for like 30 seconds.
So I said once that bacteria gets into the host, and that bacteria gets broken down, it's going to release its lipopolysaccharide or lipooligosaccharide. And the portion that is actually going to provoke the inflammatory response is the lipid A. The lipid A. And that lipid A is a PAM, is a pathogen-associated molecular pattern that will be recognized by the white blood cell, particularly the macrophages, because the macrophages have toll-like receptors that can recognize the PAM. And when the macrophages recognize that lipid A, what will happen? They will be activated and they will release interleukin 1 and TNF alpha and interleukin 6 that will cause vasodilation, that will increase vascular permeability, that will lead to symptoms and signs of shock. Do you get that, Dr. Moravia?
Yes,
So
yes, I heard that.
the shock that is going to come now is not cardiogenic shock. It is not obstructive shock, but endotoxic shock, which is a form of septic shock. Endotoxic shock, because it is an endotoxin that is causing that. And what is endotoxin? Lipid A. Where is the lipid A coming from? Lipopolysaccharide or lipooligosaccharide, which makes up the outer leaflet of what? Of the outer membrane of the bacteria. In fact, if that is the only thing we know about the outer membrane, we are okay. The other ones are not important. The embedded proteins, fine, contains porine, which allows for exchange of materials. Maybe like you want to allow enzymes that have been produced from the cell to move out, to travel to other part of the bacterial cell. They can pass through the porine. Toxic waste, they want to move out, they can pass through the porine. The inner leaflet contains phospholipids, yes. That's because it doesn't have any clinical stuff that we can discuss. The most important one is the outer leaflet that contains LPS or LOS. And what are the components of the LPS or LOS? We talked about lipid A, O polysaccharide or O oligosaccharide, as well as the O antigen. And the one that is worthy of note there is the lipid A, which is an endotoxin that can stimulate production of pro-inflammatory cytokines, including interleukin 1, interleukin 6, TNF alpha, that can cause vasodilation, increased vascular permeability, and all those things that will drive inflammation. You guys can see the application of our knowledge of pathology now. We know how acute inflammation occurs. You know the changes in inflammation. We know. So we can explain how those interleukins that are being elaborated can go to shock. Because by the time you start releasing interleukin 1 and TNF alpha, and you're having vasodilation, and in addition to that vasodilation, there's increased vascular permeability, what will happen? Fluid is going to move outwards from the vessel into the tissue. And what will happen to the into the cells in between the tissue? And what will happen to the organs? They will not be able to receive adequate blood. And what will happen? Shock. Because that's the pathophysiology of shock. Inadequate perfusion. That is it. But what is driving that shock now? Infection. That's why I call it endotoxic shock, because it's from infection elaborating endotoxin in form of lipid A. That is it. So the next one after the outer membrane is the periplasm. The periplasm is the space between the outer membrane and the cell membrane. So the space between the outer membrane of a bacteria and the cell membrane is a periplasm. The periplasm, what it contains is just to store hydrolytic enzymes. That is it. What hydrolytic enzymes are we talking about particularly? The beta-lactamases. The beta-lactamases. And we should remember that it will be present only in gram-negatives, because we have said it that the periplasm is a space between the outer membrane and the cell membrane. And we have said it that it is only gram-negatives that have outer membrane. So the periplasm is only found 2 in gram-negative. And it's a site to store hydrolytic enzymes like beta-lactamases. The beta-lactamases. This is very important too, because the presence of beta-lactamase within a bacteria can make that bacteria resistant to antibiotics. We are going to talk about it later, so you guys will understand it very well. Let me just keep quiet a little there so that I don't cause confusion. This beta-lactam, they are antibiotics.
Dr. Nabawale, is this periplasm, is it shown like in that diagram you showed us?
Yes, it is there. It is there. So this is it. So you can see that the one on top is the outer membrane. you can see the cell membrane, the cytoplasmic membrane beneath. Can you appreciate that, Dr. Antonia?
Yes.
So you can see
Yes.
the periplasmic space here. You can see it in between the outer membrane and the cell membrane
Cytoplasm.
and the cytoplasmic membrane, which contains the hydrolytic enzymes like our beta lactamases. So beta lactamase is very good for the bacteria, so that by the time the 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 person use, if that bacteria is exposed to antibiotics, particularly beta lactam antibiotics, you have the beta lactamases that are there to hydrolyze that antibiotic so that it doesn't work on the bacteria. So where did they store that hydrolytic enzyme? The periplasmic space. So after the periplasm, the next thing is the cell wall. Before you get to cytoplasmic membrane, which is the most inner portion, you get to the cell wall first. And I've mentioned it briefly that cell wall contains what? Peptidoglycan. Cell wall contains peptidoglycan. Cell wall is present in both gram positive and gram negative. So spore is present in only gram positive. Outer membrane is present in only gram negative. Cell wall is present in both of them, but they have different features. The cell wall of gram positive is very thick, while the cell wall of gram negative is not thick, it's very thin. And that explains their differences in terms of gram staining. We are going to talk about it. So don't forget, the cell wall of gram positive is thick. It has thick peptidoglycan cell wall. The cell wall of gram negative is thin. It is thin because the fact that the peptidoglycan component there is very little. Yes, so you can appreciate that in the diagram. You can see you have the cell wall here. You can see the cell wall. The layer is about 3. Can you guys see the cell wall? This one. Can we see it? 3 layers.
Yes, we can.
Okay, but out of the gram negative, we can see there is only 1 layer thick. So the cell wall of gram negative is thin compared to the cell wall of gram positive, which is very thick. And what is the component of that cell wall? Peptidoglycan. When we say peptidoglycan, what do we mean? Peptide linked to carbohydrates. So the glycan is a carbohydrate, the peptide is a protein. So what is the carbohydrate component? N-acetyl glucosamine coupled with N-acetyl muramic acid. Please let us write it somewhere if we can note it. The glycan component is actually N-acetyl glucosamine and N-acetyl muramic acid. This is not stuff. It's actually something we should know, because we'll appreciate that as we move. So N-acetyl glucosamine and N-acetyl muramic acid, they make up the glycan component, while the peptide is just sequence of protein. Peptides.
Sorry, Dr. Babalo, it's really noisy. I don't know if we can all mute our mic. We're finding it very difficult to understand what he's saying.
Paulette, you can also mute people when we inadvertently leave our mics on, because sometimes it's a mistake. The Paulette can also weigh in to mute us.
I actually do that, but some people still keep on putting their mics on. So
Okay, all right, thank you.
Okay, so we said the cell wall is made up of peptidoglycan, the peptide component and the glycan component. The glycan component is a carbohydrate. So what are the carbohydrate moieties? N-acetyl glucosamine and N-acetyl muramic acid. Attached to that N-acetyl muramic acid are peptide chains. They are peptide chains. So they all come together and they form the peptidoglycan. What I've said is very important to understand cell wall synthesis, because in the process of synthesizing the cell wall, you need to deposit the carbohydrate moieties, and you also need to deposit the protein component, the peptide component, and you need to cross-link that peptide. You need to cross-link those peptides. Sorry, I don't have a diagram. Just imagine that you have a line and you have sticking up those lines. That's those sticks that are sticking up those lines. They need to be interconnected so that they can stay together. Yes, so that is the cross-linking of those peptide side chains that are emanating from the carbohydrate moiety. And what carbohydrate moiety are we talking about? The N-acetyl uramic acid. So the enzyme that does that is called transpeptidase. Transpeptidase is the one that will cross-link those peptide components of this peptidoglycan cell wall. Now, and the transpeptidase, it's more like a broad family. We have what we call penicillin binding protein. Penicillin binding protein are enzymes. Please don't get confused. When we say penicillin binding protein, enzymes are protein. Proteins are enzymes, yes. So penicillin binding protein is an enzyme that has transpeptidase activity. Penicillin binding protein is an enzyme that has transpeptidase activity. That is, it can cross-link protein in this peptidoglycan cell wall. Beta-lactam, they are antibiotics. They have ability to bind and destroy or inhibit the penicillin binding protein. That penicillin binding protein are actually present within the bacteria. If you administer the beta-lactam antibiotics to an individual having an infection, that beta-lactam antibiotics is going to bind to the penicillin binding protein in that bacteria, so that the transpeptidase activity of that penicillin binding protein is lost, so that there's no way that bacteria can cross-link its peptide side chain. And if it cannot cross-link its peptide side chain, it cannot form its cell wall. It will eventually die. Let me stop there. I'm going to explain it again because I'm going to get to a point I'm going to talk about it. So, the cell wall contains peptidoglycan, which contains a sugar backbone, which is the carbohydrate that I've talked about, N-acetylglucosamine, N-acetyluramic acid, that is attached to peptide side chain. Where is the peptide side chain attached to? N-acetyluramic acid component. And what cross-links those peptide side chain? Transpeptidase. Transpeptidase. And I've said penicillin binding protein is an example of a transpeptidase. So we have to keep that. What's the function of the cell wall? The cell wall is just to ensure that that bacteria is supported mechanically. It is strengthened mechanically, it is rigid, and it does not succumb to osmotic pressure. That is, when you place that bacteria, maybe it finds its way into an hypotonic solution, and you have water entering that bacteria, it doesn't burst because of the fact that the cell wall can help it withstand osmotic pressure. So because of the cell wall containing the peptidoglycan, the bacteria is very rigid, it is very strong, and doesn't get busted, in response to osmotic force or osmotic pressure. Don't forget that. Then the last one, which is the innermost layer, is the cytoplasmic membrane. This one that we are seeing here. So the cytoplasmic membrane just contains three things. That's just what I just want us to know. It is made up of phospholipids, yes, just like we can see here. These are the phospholipids. You can see the heads of the phospholipid moving in opposite direction. So some are moving inwards, some are moving outwards. But their tail, they are both moving inward towards each other. So this is the way the phospholipids are arranged. But what are those things that are present within that cytoplasmic membrane? Three things: oxidative enzymes, like maybe peroxidase, oxidase; transport enzymes, like sodium potassium ATPase form; and lipoteichoic acid. So this lipoteichoic acid actually spans from the cytoplasmic membrane to the exterior. So we can see the lipoteichoic acid here. It's only present in gram-positive, though. It's only present in the cytoplasmic membrane of gram-positives, the lipoteichoic acid. So it spans from the cytoplasmic membrane to the exterior. Can we see the lipoteichoic acid here for this gram-positive?
Yes.
Yeah. So now that's like I said, one test. So the lipoteichoic acid is just the equivalent of the lipid A for gram-negative. It can also activate the macrophages so that they will release interleukin 1, interleukin 6, TNF alpha, that will lead to endotoxic shock. Lipoteichoic acid. You find it only in gram positive, just like the way you find lipid A in gram negative. So what are the three things that we said is present in the cytoplasmic membrane? Lipoteichoic acid, oxidative enzymes, and transport enzymes. So oxidative enzymes like the oxidase, the peroxidase, and other enzymes that can mediate the oxidative reaction. Then transport enzyme that can allow entry, catalyze entry into the cell, like sodium potassium ATPase pump. There are also some transport enzymes that we are going to talk about too later. Those are the basic composition of um, the the cytoplasmic membrane. Then another thing I have to mention before I go for the cytoplasmic membrane is the fact that embedded within it, we have penicillin binding proteins. So what did I say is function of penicillin binding protein? Anybody? Penicillin binding protein.
The transpeptide that cross-links the the glycans to the peptides. Yeah.
The peptide
Okay.
side chains.
Yeah, so
Cross-link
it doesn't actually cross-link the glycan to the peptide. No, it cross-links the peptide to themselves. It's the peptide that it's crossing, not glycan
Okay.
to the. Do you get it now? So
Yes, no, the N-acetyl muscarinic and is it muscarinic? Muranic and the peptide that crossing. I thought that's what you said. It joins them to the peptide.
yeah, so you have the carbohydrate, which is the N-acetyl glucosamine and N-acetyl muramic acid. So spanning side chain that is coming out from that N-acetyl muramic acid is what? The peptide side chain. Now, each of those side chain, you need to be connecting them together. You cross-link them. So you are cross-linking the individual peptide side chain. And what does the cross-linking? Transpeptidase. Trans, across. Cross-linking, across. Transpeptidase. An enzyme that catalyzes the cross-linking of the peptide side chain. What is the protein that has this enzymatic activity? The penicillin binding protein. Where do you find them? The cytoplasmic membrane. So it means that cytoplasmic membrane has penicillin binding protein that allows for cell wall synthesis. Does it make sense to us?
Yes, but if you can repeat.
So we said
One more time.
we said part of what we find in the cytoplasmic membrane is what? Penicillin binding protein, right? In addition to the oxidative enzymes, the transport enzymes, as well as the lipoteichoic acid. So we have penicillin binding protein enmeshed within that cytoplasmic membrane. We said penicillin binding protein have transpeptidase activity. So transpeptidase means you are cross-linking peptide side chain. Peptide side chain, they are attached to carbohydrates. What are the carbohydrates? N-acetyl muramic acid and N-acetyl glucosamine. So N-acetyl muramic acid and N-acetyl glucosamine attached to a protein. What do you call that? Peptidoglycan. The peptide and the glycan. Glycan means carbohydrate. What are the carbohydrates? N-acetyl glucosamine and N-acetyl muramic acid. So if you have a complex of N-acetyl stuff and the peptide, you call them peptidoglycan, and that is the composition of the cell wall. So part of the process in synthesizing that cell wall is for you to cross-link those proteins. What is the enzyme that does it? Transpeptidase. So what structure have that transpeptidase activity? Penicillin binding protein. Where do you find it? The cytoplasmic membrane. Does it make sense to us now?
Yes.
Okay. So
Yes, thank you.
yeah. So let me just so the penicillin binding protein, which is the transpeptidase, is located in the cytoplasmic membrane.
Okay.
And then it cross-links the peptide side chains,
Okay.
which bind with N-acetyl muramic acid.
Yes.
Okay, so this process of binding with N-acetyl muramic acid, which also binds with N-acetyl glucosamine,
Okay.
is what now sheds off the cell wall.
That's what forms the peptidoglycan. That is That means you are trying to form peptidoglycan that makes up the cell wall. That is it.
Okay.
Because
Okay.
we
So
said
it's
that
just like pushing. So it's like pushing. I'm just trying to walk. So it's like pushing the stuff up. So the cell membrane is forming the stuff and pushing it up to form the cell wall.
exactly. So you know that if you want to synthesize something, you need enzymes. So the
Yeah.
enzymes that form the cell wall are just from the cell membrane.
The
That's
cell
what it
membrane.
means.
Okay. Okay. Okay.
So that
Fair
means that
enough.
if I want to kill off a bacteria, one way I can kill off a bacteria is to what? Inhibit the cell wall synthesis. Do you guys
Okay.
get that? So if I want to inhibit the cell wall synthesis, what can I do? Give an antibiotic that can bind to the penicillin binding protein so that I will inhibit that penicillin binding protein. An example of that antibiotic is what? Beta-lactamate. Beta-lactam antibiotics like penicillin. So that's why we call it penicillin binding protein, because it can bind to penicillin. Penicillin is a beta-lactam antibiotic. So penicillin will now go to bind to its protein, which is penicillin binding protein. So by the time penicillin binds to that protein, it will inhibit that protein so that the protein will not be able to um, exert its transpeptidase activity. So there's no way it can cross-link the peptide side chain to form the complete peptidoglycan that will form the cell wall. Do you guys
But
get
when
it?
you were calling, yes, but when you were calling the enzymes in the things that are contained in cytoplasmic membrane, you said oxidative enzymes like oxidases and peroxidase, transport enzymes like the sodium potassium ATPase,
Okay.
but you did not call the transpeptidase.
Yes. So I said penicillin binding protein, but you
Okay,
already
yes,
know, that penicillin binding protein has transpeptidase activity. Here you get. So that's just the addition, Dr. Moravia. Do you get it now?
okay,
So that's
okay,
just an
okay.
omission on my part. Yes. Thank you very, very much for that. So we have discussed uh the whole structure of the atypical bacterial cell. That is everything. So
Dr. Babawale,
yes, Dr.
sorry,
Paulette.
please can you list the components of cytoplasmic membrane again? The penicillin binding protein, oxidative enzymes, transport enzymes, and what again?
So the oxidative enzyme, the transport enzyme, the penicillin binding protein, the penicillin
Okay.
binding protein, the transport enzyme, the oxidative enzyme, then lipoteichoic acid. Those
All
four
right.
things. But
Thank you.
the one I want us to hold majorly is what? The penicillin binding protein. Yes. Most likely they will not ask us that where do we find lipoteichoic? Nobody will ask us that. If it's going to come in a question, they will just ask you what would it induce? It can cause endotoxic shock because it can trigger elaboration of your interleukin 1 and TNF alpha. They will not ask for location. It's very unlikely. Even all these location, we are just saying it just for saying sake. They really test location of all these things. But it's just important for us to appreciate the clinical application of all this stuff that we have been talking about. For example, the utilization of penicillin in treating bacterial infection as an antibiotic is because of the fact that the bacteria have penicillin binding protein that penicillin can inhibit. That's so let's just run through them. Should I just move or I I go through them once more? I know it's not easy for us. Actually, I'm kind of in a
Once
very
more, please. Sorry.
Please, can
There's
you
a
go
lot
through
of
it?
interruption. Once more,
okay.
Please
please.
go through it once more. Thank
Thanks.
you.
So should we start from up to down, the appendage downwards, or should we start from cell envelope only?
From the appendages.
Ah, Doctor,
Hello,
Sorry.
Chief. Hello,
There was
Chief.
a lot
Please,
of
Oh,
interruption,
I'm
really.
listening, Doctor Obaje. Is that Doctor Obaje?
please, can you guys hear him?
Yes, we can hear you, Doctor
Yes.
Obaje.
it's-
I can't network with this now for crying out loud.
start from the appendages,
We can
please.
hear
Thank
Okay,
you.
so let's start again. The appendages. Appendages are just additional structure that are attached to the cell. Three of them: the flagellum, the pilus, and the fimbriae. So the flagellum is made up of protein only, and it's for movement. So when we say an organism is flagellated, it can move. That is it. It's an appendage. It's an additional structure, and it's whip-like. The next one is the pilus. Pilus and fimbriae, both of them are made up of glycoprotein, not only protein. So they have carbohydrate attached to that protein, glycoprotein. What's the function of the pilus? The pilus is for conjugation. What is conjugation? Ability for bacteria to come together and transmit genetic material from one cell to another cell across a bridge. Across a bridge. So they will come together. That sex pilus is going to do formation of a mating bridge. So that bridge is going to connect those bacterial cells together so that they can transfer genetic material to each other. That genetic material can be anything. Like I said earlier, it can be anything that will, maybe something that will make them resistant to antibiotics, or something that will allow them metabolize some particular metabolite or molecule, or something that will just make them survive. It can be anything. It can be anything. Just know that pilus is for exchanging genetic material. That is it. Through the process of conjugation. Then fibrae is also made up of glycoprotein. And what does it do? It's just for attachment to surfaces. Attachment to surfaces. Attachment to surfaces. So that they can colonize that surface and they can cause infections. I gave an example of E. coli. So if they ask us now, which of the following cellular components is E. coli using to cause UTI? You pick fibrae, because E. coli will use its fibrae to attach to the epithelium of the genitourinary tract, the uroepithelium, that is the epithelium lining the urinary tract. That's what we call the uroepithelium. It's because E. coli has that fibrae. That's how it can attach to that epithelium and cause urinary tract infection. Do we get it? We are going to explain the other ones too.
Yes.
So that is that about the appendages. That is all. Then we'll go to specialized structure. We said the specialized structure that we're talking about is spore. What is a spore? Spore is a form that the bacteria assumes so that it can survive. It can survive when the environmental conditions are very harsh. For example, when the bacteria is outside the host, is in the environment, maybe in the soil. In the soil, there's no water. In the soil, it can be exposed to sunlight and it can dry up. And all these things can denature the DNA. It can kill off, it can destroy the DNA and the bacteria will die. So what would the bacteria do so that it can protect itself from all these things? It can assume that spore form. So, and we said spore is made up of so many things that protect the bacterial genome from the outward to the inward. So outward, the exosporum. We talked about the next one, which is a coat. What is the component of that coat? Keratin-like substance. So we have the keratin-like coat. After the coat, cell wall. What is the major constituent of cell wall in bacteria? Peptidoglycan. The next thing is the genome, the DNA. But what surrounds that DNA? Dipicolinic acid. That is what prevent that DNA from becoming, prevent it from getting damaged from heat exposure. So that spore form is going to allow that bacteria to survive, and it's going to protect it from heat, from chemical, and from dehydration. From heat, from chemical, and from dehydration. Because of all these specialized constituents that we have talked about, the keratin-like coat, the dipicolinic acid, the thick peptidoglycan cell wall, all of them are protecting the DNA. Okay. Do we understand that? Spore.
Yes.
Yeah,
Yes.
yes.
The next, the
Thank
cell
you.
envelope. So a cell envelope is just like conglomeration of so many things that surround that bacteria. So from outward to inward. So we talked about the capsule. That's the one that is the outermost one, the capsule. So not all the bacteria have capsule. We said it is only bacteria that are encapsulated that we can say they have capsule. And what is the function of the capsule? Is to make it resistant to phagocytosis. Yes. So what is the composition of that capsule? Polysaccharide. So we say polysaccharide capsule, polysaccharide capsule. It is a polysaccharide capsule. In fact, that capsule is antigenic, that we can obtain a capsule of a particular bacteria and create vaccine from there. I know we know that from immunology, but I'm still going to say it so that it will stick. I'm not going to talk about it now. We'll talk about it later because we'll get to a point where we'll be talking about the capsule. So don't forget, the capsule is made up of polysaccharide, and it's there to prevent phagocytosis, so that the neutrophils, the macrophages will find it very hard to digest that bacteria. Any bacteria that is not encapsulated, they can easily phagocytose and digest it. What's the next layer after the capsule? The slime layer. Not all the bacteria have it too. Any bacteria that has this slime layer can form biofilms. So biofilm is just like aggregate of bacteria adhering to a particular cell surface, attached to that cell surface with the aid of extracellular matrix component. So you just picture it as colonies of bacteria adhering to a surface, and we said that surface can be anything. Majority of them are foreign substances that are introduced into the body, like prosthetic heart valve, like prosthetic hip implants, like cornea lens, all these contact lens that we attach to the cornea, ventilator. So somebody that is exposed to ventilator for a long time, you can have bacteria colonizing those ventilator tubings, and from there they can travel to the lungs and cause pneumonia. We'll get there. So that is biofilm. Colonies of bacteria adhering to a surface, and they achieve that by the aid of their slime layer, which is made up of polysaccharide, just like the capsule. Then after the slime layer, we now start talking about the outer membrane. So you guys might not even need to know the arrangement. It's not necessary. Just be able to appreciate the function and the composition. That is it. Outer membrane, we said three things. It has the outer leaflet, the inner leaflet, and in between the leaflet, you have the embedding protein that contains sporine. The outer leaflet is the one that I want us to always remember. What does it contain? It can contain lipopolysaccharide or lipo-oligosaccharide. Which bacteria did I say is made up of lipo-oligosaccharide? Anybody?
Bacteria and its
Bacteria. You see, you guys already know the details. I don't even need to be going back again. No. That is it. Those are the take home that you guys need to know. Neisseria is the one that contains the LOS. Other ones contain LPS. E. coli, Kleb, Citrobacter, and all those ones, it is LPS. But Neisseria is LOS. Very important. What is the composition of that LPS? What is the individual subunit? It's made up of lipid A, O polysaccharide, or O oligosaccharide, depending if it's LPS or LOS, and the O antigen. Forget about the O antigen and the O polysaccharide, or forget about it. The one that you need to remember is lipid A, because lipid A is capable of causing endotoxic shock, because lipid A can activate the macrophages. Lipid A is a pathogen-associated molecular pattern that can be recognized by the toll-like receptors of the macrophages, and it will make the macrophages elaborate pro-inflammatory cytokines like interleukin-1, interleukin-6, TNF-alpha. And you guys remember from pathology that once you elaborate these cytokines, you are playing with inflammation. And when there's an inflammatory response, there's extravasation of fluid from the intravascular compartment. That is why when you define shock, clinically, they will define shock as reduced effective circulatory blood volume. That is, the blood volume that's supposed to be within that vessel, not the one in the tissue. No, the one that has extravasated into the tissue is no longer effective. So in someone that have elaboration of this interleukin 1, 6, and the TNF, there is going to be increased vascular permeability that will make blood go out of the vessel into the tissue, causing edema. That's why edema is one of the cardinal features of inflammation. When there's edema, you have reduced blood volume. We have reduced blood within the intravascular space. Most of them they have gone extravascularly. So there's little effective blood volume to go to the organ and perfuse them. So we can have AKI. The person can have ARDS. The person can have cerebral perfusion that is reduced, and the person will have features of sepsis. Why? Because what is mediating those changes is what? Endotoxin. Endotoxic shock. Because endotoxin is elaborating inflammatory cytokines that is increasing vascular permeability, causing fluid to leave the vessel, and reducing perfusion to the organs. Because what is shock? Reduce perfusion to the organs. So what type of shock is this? This is not the heart failing. This is not PE or obstruction. This is not anaphylactic. But what? septic shock from infection. What is infection? Elaboratory. Lipid A. What is lipid A? Endotoxin. So we can call it endotoxic shock too, because of the TNF1, interleukin 1, and interleukin 6. Do you understand that? Hello, my people.
Yes, we understand. Yes.
After the outer membrane, we go to
Yes.
the periplasm. Periplasm is a space between the outer membrane and the cytoplasmic membrane. What does the periplasm does? It stores the hydrolytic enzyme. Which hydrolytic enzyme? Beta lactamases. Beta lactamases are synthesized by the bacteria. How? They synthesize it in order to protect themselves from beta lactam antibiotics, penicillin. Now imagine that you give somebody penicillin and the person has bacterial infection. That penicillin is going to go to that bacteria and what? Bind to the penicillin binding protein in that bacteria, so that it can inhibit the penicillin, so that it can inhibit the penicillin binding protein, so that the person will not be able to form the cell wall. But the bacteria already have something in stock to fight that. So within their periplasmic space, they have kept some hydrolytic enzyme, particularly beta lactamases, that can hydrolyze beta lactam antibiotics like penicillin. So that is the basis of developing antibiotics like before I get there, do you guys understand what I've said now about the beta lactamases?
Yes.
That bacteria themselves have hydrolytic enzyme that can hydrolyze beta lactams. What are beta lactams? They are antibiotics. Example of beta lactams, penicillin. What is the target of penicillin in the bacteria? It binds to the penicillin binding protein so that it can inhibit that protein. Please, let's mute our mics. But with the knowledge of molecular biology, we are now able to now checkmate the activity of that bacteria. Okay, fine. You bacteria, you guys have now started developing hydrolytic enzyme like beta lactamase within your periplasmic space. We know what to do. So what we now do is that in combination with our penicillin, why don't we add another thing that can inhibit beta lactamases in the bacteria? So that is the basis for developing combination like augmentin. So you have amoxicillin, clavulanic acid. So amoxicillin is a penicillin component. The clavulanic acid is the beta lactamase inhibiting component. So if the bacteria have beta lactamase within their periplasmic space, there's no problem. We already have clavulanic acid added to our penicillin, and that clavulanic acid is going to destroy that hydrolytic enzyme in that periplasmic space, which is beta lactamase. So it will allow our penicillin to work. Which penicillin are we talking about now? Ampicillin. That's the basis for developing piperacin tazobactam. Tazobactam will inhibit the beta lactamase. It will allow the penicillin to work. What is the penicillin? Piperacillin. Please, guys, hope I've not confused us with what I've said.
No.
I think you just confused us just now.
I was confused.
What Dr. Kenneth said. Okay. So, okay, let's go back again. Let's. My battery is almost dead, but let me relax and say it again. Now,
Thank you.
we know that in the bacteria we have penicillin binding protein, Abi. So penicillin binding protein is within the cytoplasmic membrane of the bacteria. And what does it do? What it does is that it helps to form the cell wall of the bacteria because it has transpeptidase activity. It can cross-link the peptides to form the cell wall. Now, the bacteria at the same time, in addition to having that penicillin binding protein, they have beta lactamases. Beta lactamases are enzymes that can hydrolyze beta lactams. What are beta lactams? They are like penicillin. They are penicillins. Penicillins have beta lactam ring. So the bacteria have I want to talk like Dr. Babaide now, like an hammer that can knock off all those penicillin. So example of that hammer is what? Beta lactamase, because penicillin is a beta lactam. So we have beta lactamases that can destroy beta lactams like penicillin. That armor is beta lactamase. Where do we find it? Periplasmic space in the bacteria. If we give somebody penicillin, that we want to use penicillin to kill off a microbe, what that penicillin, which is that beta lactam, is going to do is what? To bind to penicillin binding protein. Just imagine that penicillin is binding to penicillin binding protein. It will inhibit that protein. And if you inhibit the activity of penicillin binding protein, what will happen to the bacteria? It will not be able to synthesize the cell wall. But bacteria, they now have something. They have what we call beta lactamase, that armor, so that when that penicillin is coming, they will knock it off with their beta lactamases, hydrolytic enzyme that can hydrolyze penicillin, so that penicillin will not get to bind to the penicillin binding protein. Do we get it up to that level?
Yes. Thank you.
So the basis of me now saying piperacin, tazobactam is now that, oh, we know that there are some bacteria that have beta lactamases, which are something that can inhibit penicillin. Does it mean that penicillin will no longer be working? No. Penicillin must work regardless of if the bacteria has beta lactamase or not that can hydrolyze the penicillin. So what do we now do? We add something in combination with the penicillin. Please, you guys should note that penicillin is actually a broad name. We have penicillin V, penicillin G, fine. But ampicillin is also a penicillin. Hope you guys agree with me that ampicillin is a penicillin.
Yeah.
We are going to get there when we are talking about antibiotics. It's in this same microbiology. It is down. Now, if you want to give somebody ampicillin and the bacteria have beta lactamase, ampicillin will not work. So instead of giving ordinary ampicillin, we add it with a beta lactamase inhibitor. So we add, in addition to ampicillin, add a beta lactamase inhibitor that will allow your ampicillin work. So what is an example of that beta lactamase inhibitor? Clavulanic acid. So it means that once you give somebody a combination of ampicillin, clavulanic acid, once that combination gets to that bacteria, the first thing is that the clavulanic acid is going to inhibit the beta lactamase, so that in the bacteria, so that there's no beta lactamase in the periplasmic space in the bacteria to inhibit or to degrade or to hydrolyze the penicillin, which is ampicillin, so that ampicillin will be capable to work. And how will ampicillin work? Ampicillin is a penicillin. How do they work? They bind to penicillin binding protein in the bacteria. So when it binds to the penicillin binding protein, it will inhibit that penicillin binding protein, and they cannot exert their transpeptidase activity again, and they cannot participate in the formation of the peptidoglycan cell wall because they can no longer cross-link the peptide chain of the peptidoglycan. And you guys remember that peptidoglycan is an essential component of the cell wall. Do we understand now?
Yes. Thank you.
Yes, we do.
Okay. So we've talked about the periplasm. That's all about it. It contains the beta lactamases. The next one is the cell wall. We've talked about cell wall already. Cell wall contains peptidoglycan, and it's present in both gram positive and gram negative. Sorry, yeah, gram positive and gram negative. It's just that in gram positive it is thicker. In gram negative it's very thin. That is it. What's the function of the cell wall? For rigidity and for osmotic support. For rigidity, mechanical, and osmotic support. So after the cell wall, what's the next layer, which is the innermost layer? The cytoplasmic membrane. 4 things that we find in the cytoplasmic membrane: lipoteichoic acid, the oxidative enzyme, the transport enzymes, and our penicillin binding protein that we've been talking about. Penicillin binding protein is there so that they can synthesize the cell wall. The transport enzymes are there, like sodium potassium ATPase form. You guys know that sodium potassium ATPase form is present in almost all the cell membranes. It has to be there. It's a transport enzyme because it will allow for transfer of sodium and potassium. Oxidative enzymes like peroxidase and all those things to maintain normal metabolism in the bacteria. Then lipoteichoic acid, which is only present in the cell membrane of gram positive. acting like lipid A because it can also stimulate release of interleukin 1, TNF alpha, and interleukin 6 that can drive endotoxic shock or septic shock. Do we understand, guys? Do you understand? I can't believe we spent the
Yeah.
entire stuff discussing one page. Oh. Hey. Can't continue like this. Oh. So, let me see if before my battery dies, let's go to the gram staining. So, the gram stain is the first line laboratory test that we use in bacterial identification. That's the most basic. In fact, it is the most basic. That's the first thing that you do to classify a bacteria. And it actually depends on the property. It's the principle for this gram stain is actually relying on the chemical and the physical property of the gram of the cell wall. So if the cell wall is thick, it's likely to be gram positive. If it's thin, it will be gram negative. I will explain. So what happens in the gram staining technique? What do? Just to rush us through the process, the first thing is you want to apply a premise stain to the bacteria. You apply a premise stain. What's the premise stain? Crystal violet. Crystal violet has violet or purple color. After you apply that premise stain, you add some other things like iodine. Let's forget about iodine for now. The next thing, you add a decolorizer. Now, the purpose of that decolorizer is to wash off that premise stain, which is your crystal violet. You want to wash it off. But if the cell wall is very thick and it has abundant peptidoglycan, they are going to take that crystal violet. They are going to take it. They are going to take a lot of that crystal violet, such that your what's it called? Your decolorizer will not be able to wash it off. Usually you use alcohol to decolorize. But if you are dealing with a gram-negative bacteria, fine. They will take little of the crystal violet because they have thin peptidoglycan cell wall. They will take little of it. By the time you apply your decolorizer, your alcohol, that alcohol is going to decolorize and wash out all the stain that it has taken. So it will look neutral. But for gram positive, it will have taken a lot of that crystal violet because it has abundant peptidoglycan wall. And by the time you add your decolorizer, it's going to wash out a little. You still have abundant crystal violet being present within that cell. So after you have applied your decolorizer, the final thing is now to apply a counter stain. The counter stain that we usually use is safranine. It has a red color. So by the time you add your counter stain to your gram positive, because your gram positive is already withholding your premise stain, which is crystal violet. By the time you add your counter stain, it's going to take that counter stain, but it's still going to appear purplish. Why? Because your primary stain is darker than your counter stain, which is red. So it will still retain the color of your primary stain. So gram positive will appear violet or purplish. But for your gram negative, because your decolorizer has washed out your primary stain, which is the crystal violet, it is neutral. By the time you add your counter stain, it's going to take that counter stain and it's going to assume the color of the counter stain. So they will appear red, safranine, red. Do you understand that process of gram staining? Does it make sense to us?
Yes. Yes.
So it is the thickness of the gram of the cell wall that will determine what you eventually see. A very thick cell wall is going to retain your primary stain, which is crystal violet. It will be resistant to the activity of your decolorizer. So it will appear purplish or violet color, purple. But your gram negative has a very thin peptidoglycan cell wall. It's not going to absorb the primary stain, and by the time you decolorize, all the little primary stain that has absorbed will be washed away. By the time after you now decolorize, you now apply your counter stain, it's going to take that counter stain only, and the counter stain is red. That is why gram negative will appear red. That is the technique of gram stain. Very easy. So what are the microbes that don't respond to this gram staining procedure? You can't gram stain them. Before I leave the gram staining process, you guys should remember that Once you are done with your gram staining stuff, you need to visualize it with a microscope, a bright field microscope, a bright field microscope. Now, what are the microbes that we cannot gram stain? We have about 4 categories. So the first category, we call them the spirochetes. The spirochete because they appear spiral. They appear spiral. Example, the treponema and leptospira. You guys should not, you guys don't worry yourself about these things. By the time we go deep in this microbiology, all these things they will come to you spinal level, like you know them spinally. This treponema and leptospira, they are under the class of spirochetes. Once you hear spirochetes, you are thinking of leptospira and treponema. So treponema is where you have syphilis. So syphilis, treponema pallidum. Leptospira, that's leptospirosis. Don't worry about it, we'll get there. We'll get there. So both of them, they are too tiny to be seen by a bright field microscope. So that is why even if you can see them, you cannot see them. So the best technique to see these guys is to use a dark field microscopy. Use a dark field microscopy. It is sharper than your bright field. You'll be able to appreciate these guys. What's the next one? Your mycobacteria. Mycobacteria tuberculosis, Mycobacterium leprae. Mycobacteria generally, you cannot gram stain them. Why? It is because their own cell wall contains mycolic acid. So instead of containing the typical peptidoglycan, it contains mycolic acid. Mycolic acid is a lipid. It is very thick, it is waxy, and it does not readily pick up any stain. So guys, let's not imagine that you try to gram stain mycobacterium and the mycolic acid does not pick up crystal violet. It does not pick up safranin. What are we going to see on the gram stain? Are we going to see anything?
No.
No, we're not going to see anything. That is why you don't gram stain mycobacterium. You do another stain that we are going to talk about. Because of the fact that it has cell wall that contains mycolic acid, which is a lipid that is there. And that lipid doesn't readily take up those stains. Mycoplasma, ureplasma, the plasmas, those guys, they are like together. They have no cell wall. So there's nothing to even pick up the stain. They have no cell wall. This is very important too. We must remember this attribute of these guys. Mycoplasma, ureplasma, they lack cell wall. Very, very important. So it is logical to say that we cannot use an antibiotic that inhibits cell wall in the treatment of mycoplasmal infection. So if somebody has mycoplasma pneumonia and the person is having pneumonia from that, you cannot give that person penicillin. It will not work because penicillin will inhibit synthesis of cell wall. These guys don't have cell wall. What are you inhibiting? Give something that can inhibit protein synthesis. That is why doxycycline can work in the treatment of these guys. I'm just chipping in that. Don't forget, mycoplasma, ureplasma, they lack cell wall. Lastly, we have these guys: Legionella, Rickettsia, Chlamydia, Bartonella, Anaplasma, Ehrlichia. So I think I have an acronym. Okay, yeah. So the acronym that I use is CABLE. That is if you want to remember it. To be honest, it's not actually very important. The one I know it's actually important here is Chlamydia, Rickettsia, and Legionella. Those three. We need to know them. Rickettsia and Chlamydia, they go together. Rickettsia and Chlamydia, they go together. They are like brothers. So you can just add Legionella to them. But if you remember, you want to remember the everything, the acronym is CABLE. So C for Chlamydia, A for Anaplasma, B for the Bartonella, L for Legionella, E for Ehrlichia. So the reason why we cannot gram stain these guys is because they are intracellular. When we say an organism is intracellular, it means that it stays within some cells within the host. For example, if somebody gets infected with chlamydia, chlamydia is not going to be staying inside the plasma. No. Chlamydia will be engulfed by the macrophages, so it's going to stay within the macrophages. Macrophages are eukaryotic cells. They don't have peptidoglycan cell wall. So it means that if you gram stain that kind of specimen, you need to penetrate that macrophage. Yes, because it doesn't exist outside the macrophage. So there's no way you can pick up that it can pick up the crystal violet and the saffronin stain. So you'll not be able to see it because they are intracellular. So intracellular organisms cannot be gram stained. Chlamydia in particular cannot be further gram stained because its cell wall is not well developed. Its cell wall lacks the muramic. We can see it here that there's a reduced muramic content of the cell wall of chlamydia. Yes. So you guys remember when I was saying it then that one of the components of the cell wall, the peptidoglycan, is what? N-acetyl-muramic acid. So that muramic acid component in chlamydia is very reduced. So that cell wall is not well formed. So it cannot readily pick up stain because of that too. Aside the fact that it is intracellular. So that's the word. Acronym CABLE. CABLE. C for chlamydia. And I've told us whenever we remember chlamydia, the next thing that will come to our mind is rickettsia. They go together. Chlamydia, rickettsia, chlamydia, rickettsia. We are going to appreciate that as we further go in our discussion of this microbiology. Chlamydia, rickettsia. So CABLE. C, chlamydia. A, anaplasma. You see, anaplasma, they like staying in the granulocyte. That's where they like staying. Anaplasma is for granulocyte. Granulocyte, anaplasma. You will see it as we go. Anaplasma, granulocyte. The next one, Ehrlichia. Ehrlichia and anaplasma, they are like similar too. But instead of Ehrlichia staying in granulocyte, Ehrlichia stays in monocytes. Yes, monocytes, Ehrlichia. Granulocyte, like our example of granulocyte. Anybody give us example of granulocyte? Anybody? Example of granulocyte, so that I'll know you guys are following me.
Neutrophil.
Neutrophils.
Yes.
What else? What else?
Basophil.
Basophil. Thank you. Thank you. So those guys are granulocyte. Anaplasma like staying in those guys. Ehrlichia would rather stay in the monocyte, not granulocyte. So CABLE. C, chlamydia. A, anaplasma. B, Bartonella. Bartonella is the one that causes catch disease. Bartonella and Selly. People that have done dermatology, they will remember that. L is a Legionella. Legionella, we must know it. If that guy causes a lot of things, we must know it. E is Ehrlichia. E is Ehrlichia. And we have said it that Ehrlichia is the one that stays in the monocytes. So all of these guys, they are intracellular. They stay in one white blood cell or the other. So they cannot be penetrated. Now, aside the Giemsa stain, there are other stains that we can know. I'll just try to put us through some ways we can learn some of them. Actually, it's not all these things that are important. You see, the ones that are very important there that we need to know, I think, periodic acid sniff, zenestin, Indian ink. Those ones, we cannot joke with it. You cannot go to that exam and not know what Indian ink stain is for. In fact, it's like someone is playing. You don't know what Indian ink stain is for. You don't know zenestin stain, what it's used for. It's bad. So these are stuff that we need to know. So the way I learned this Giemsa stain, so I just go back to all these atypical one, this intracellular one rather. So the first thing about Giemsa stain that I want us to know is that Giemsa stain is peculiar to staining protozoas. Protozoas like Plasmodium and Trypanosomes. So sleeping sickness, Trypanosomes. All these Trypanosomes, Miasis, Trypanosoma miasis, Trypanosomiasis, and the others, they are protozoas. So Trypanosome brucei, Trypanosome gambiens, all those things. Trypanosome cruzi. They are all protozoans. Malaria caused by Plasmodium is a protozoa. And what you typically use to stain protozoa is Giemsa. You have known that fact like that. Now, let's go back to all these intracellular organisms. Let's try to separate them. So I've said it that chlamydia and rickettsia, we try to pair them together. So if you cannot gram stain chlamydia and rickettsia, what stain can we use? You can use Giemsa. You can use Giemsa. So we have already 4 organisms for Giemsa. Our protozoans, which includes Trypanosomes and Plasmodium, and our chlamydia and rickettsia. Chlamydia and rickettsia, Trypanosomes, and Plasmodium. Now, let's go back to our intracellular organisms. We have said it that rickettsia and chlamydia can be stained by giemsa because we cannot gram stain them. What can we do? We can use giemsa stain for chlamydia and rickettsia. Which one do we now use for Bartonella and Legionella? Which one do we use for Bartonella and Legionella? Those are the ones that even follow each other in the cable acronym BLE, BL, Bartonella and Legionella. What do you use to stain those guys? Because you cannot use gram stain. What do you use? You use silver stain. Silver stain. So, silver stain will work for Bartonella and Legionella. Gram stain will work for, giemsa stain will work for chlamydia and rickettsia. Helicobacter pylori, which is the organism that causes PUD, can both be stained by giemsa and silver stain. Helicobacter pylori is the one that is central to all of them, to both of them. You can see it with giemsa, you can see it with silver stain. So it means that we can definitely say many things under the giemsa stain. Number one, we can say our protozoa, trypanosome and plasmodium. We can say our chlamydia and rickettsia because they go together, and we can add the common one, which is Helicobacter pylori. We are done with giemsa. That is one. Then we already know part of silver stain. We've said it that Helicobacter pylori is central to both silver and giemsa. So Helicobacter pylori can be stained with silver stain, and we've said it that the BL, Bartonella and Legionella, both of them can also be stained by silver stain. So we have Bartonella, Legionella, and Helicobacter pylori. Just like giemsa stain, that you must remember your protozoa. For silver stain, you must always remember your fungi. Fungi. In fact, the first thing that should come to our mind when they mention silver stain is fungi. Silver stain, silver methanamine stain, fungi. So let's go to the box for silver stain. We can see the common one, Helicobacter pylori. We can see our BL, Legionella, and our Bartonella. We can see the fungi. An example of the fungi, they have listed them. We can see that we have learned the organisms that we can stain with giemsa and silver stain just by knowing the intracellular organisms. I hope this makes. I'm going to go off anytime soon. Do you guys appreciate what I've said?
Yes.
Yes, Chief.
So these things are not very easy to memorize. That is why I'm just using that technique. Although they are not very high yield. Giemsa and no. You once for the giemsa, just essentially know your what's it called? The protozoas. For your silver, just know fungi. That's the one that they commonly.